I'm Kyle Hawkins. I am interested in doing a hackathon project on making a cellular Potts model of dental plaque, looking at the spatial structure and having pH feedback within that model. So for the biological background, dental plaque is usually modeled as a biofilm. You have a community of different bacterial species. And as far as interfaces go, you have a tooth to plaque interface, and you also have a saliva to plaque interface. Within your plaque biofilm, diffusion is how you're going to have exchange of solutes throughout it. And further, you have the development of microenvironments where in different spaces or different spatial spots, or I guess sites, you're going to have different pH values. A lot of the pH dynamics of these microenvironments are dependent on the addition of glucose substrate or lactate substrate, or sorry. So you have substrate, and usually when you introduce your substrate, you'll get fermentation. That fermentation causes a dip in your pH, and then it recovers. This curve here is called the Stephan curve. So that's the behavior you see. As far as the community of microbes, you have acid-producing species and you have base-producing species. So you have this interplay of some species prefer acidic environment, some species prefer basic environments. And I think originally when I was thinking of a problem, I wanted something that had that dynamic. So as far as my hypothesis goes, my hypothesis is that you can build these pH microenvironments, which you can see in this image here. This is what the microenvironments look like. You can build these microenvironments through a species-specific pH-dependent growth rate, kind of modeled something like this, not necessarily this form, but that's my hypothesis, that that alone is sufficient to build these microenvironments. And as far as like broader impact goes, tooth decay, cavity development is highly dependent on pH. You can see in this image here, you can see a lesion forming as you have more and more acidic environments. So that's a broader impact. You could also use this to inform different control interventions for having a more, I guess, distribution focused treatment plan rather than, I guess, number of microbes per lot. So far as modeling goes, or modeling goals go, this requires quantitative modeling, one for the pH, two for the configuration itself. I want to test if these pH-dependent growth rates are sufficient for making the microenvironments. And as far as the prediction goes, I think doing like a 3D planar plate basically is the best starting point. There might be a 2D version potentially, depending on constraints of the hackathon. And as far as broader impact, I kind of already mentioned broader impact. As far as the plan goes, basically trying to determine the metabolism of the process, get the different parameters for it, develop the pH calculation, and sort of build out the sort of wash response cycle. And then, of course, documentation, validation, and analysis. I think CompuCell3D should be very helpful for understanding the spatial arrangement of the biofilm. As far as resources, these are some papers that I've identified as being helpful. Also, I have some Julia-based code for doing the pH calculation, which I think I should be able to convert into Python. And as far as teammates go, I'm looking for people that are interested, that have knowledge about the microbiology, potentially help with parameter mining, maybe some of the reaction diffusion mechanics. if people know about demineralization, that could be an interesting stretch goal to model, and analysis, figures, and coding expertise. So that's my pitch, and thank you all for listening.
That's great. I think that would be nice to see. People have talked about doing dental biofilm modeling for a long time, but I don't know how much people have actually done with it. And so I noticed you cited the Pistoriano paper from 10 years ago or so. That's quite an inspirational paper.
Yeah,
I remember that paper very well. I think the problem usually comes about because bacteria are very small. And so probably one CompuCell bacterium probably represents 10 or 100 real life bacteria just because of the length scales. On the other hand, you can certainly get the idea of communities where one community produces a resource that another community uses, or a community produces a toxin that suppresses other communities. And so you can definitely, I think, get interesting results in terms of coexistence and patterning of different bacterial populations on a surface. So I think and I think a simple, probably 2D version of it, you could get done in a weekend. Going to a full 3D model might be a little bit more ambitious, but I think it's a really great project. And it's also something where if there are people who are looking for a project and don't have a lot of experience, that might be one where you could have people come at it from different angles and be a good learning experience for everybody.
Hi everyone, I'm Shivani and I'm from Indian Institute of Science, Bangalore, India. I am presenting this project for hackathon, basically that it's about modeling cellular heterogeneity and collective migration during EMT. So when a tumor decides to proliferate and starts to invade a secondary site, moving from a primary site, one of the main thing it do is goes under epithelial to mesenchymal transition so that it becomes invasive and migratory. Epithelial cells are those cells that have high cell-cell adhesion and less cell-ECM adhesion. Therefore, they tends to bind to a cell of same type, whereas mesenchymals are more migratory cells that have a invasive tendency and have more cell-ECM adhesion. Hence, they can break down the ECM around them, creates a pathway for the cells to starts to invade. Now, the problem that I'm dealing with is instead of defining a two distinct phenotype as epithelial and hybrid, we can consider the heterogeneity of tumor with a with a cell collection of cell of varying level of adhesion strengths, both cell and cell ECM, thus creating a heterogeneity depicting how a real tumors look like. So that it creates a leader-follower dynamics where leaders are more cells with mesenchymal phenotype having more cell ECM adhesion that breaks down the ECM, creates a pathway, and followers are more epithelial-like cells that basically adhere to the mesenchymal cells and follows them through the blood vessels. The first question is that I'm interested in is to create a dynamic adhesion strength population. And second, to see how the cluster formation due to this dynamic adhesion strength can extend can change the extent of invasive front. For example, if our clusters create a spheroid structure, will that be more invasive or will it create a longitudinal cluster? So will that be more invasive? So the basic hypothesis is that there is a continuous phenotypic switching from cell and cell ECM adhesion that creates a dynamic framework that leads to a spontaneous cluster formation. These cluster formations can be of different size and shape that can affect the extent of invasion, thus help us to study how a tumor actually invade and whether it leads to a collective cell invasion or single cell invasion or uh, molecular invasion. Second, what are the inputs and outputs? The inputs will be of course, cell-cell adhesion, cell-ECM adhesion, phenotypic switching between cells of various type, MMP secretion level because mesenchymal cells have high MMP secretion. That's why they can degrade the ECM. Next is the ECM properties. ECM properties, we can vary the stiffness and density of the ECM to obstruct the extent of invasion. The output will be the cluster formation and morphology, leader follower dynamics, invasive distance, invasive front, and extent of ECM degradation. So, what are we predicting? The model aims to predict how adhesion heterogeneity influence cluster formation. And how are we going to do this? I have a basic setup that one of my seniors has done in CC3D. So here I am going to show you one of the simulation, the basic setup that we already have is the blue one are the ECM, green is a basal membrane, and red and yellow are the two types of cells. Initially uh, for this simulation, I have given all the coupling strength, or we can say cell ECM value for two cell types completely same, which is C1 is depicting ECM here. This is basal membrane, and this is adhesion between two cells of same type, two cells of different type. So this is similar right now. So what we observe when we run this simulation is that both cell, because of same adhesion strength, have similar invasive front. They try to initially degrade the basal membrane, and that's how they enter the ECM matrix. Then they start to degrade ECM so that they can enter the blood vessels, and from that blood vessels, they finally enter from their primary site to a secondary site. This is a basic model. And if we change the adhesion strength here from 20C. If we increase this number, then the adhesion value will decrease that we have run yesterday. So if we change this number, we can have we can have two different phenotype depicting two different adhesion strength. But the question is, instead of defining two state like this, we can create an environment where the cells have a varying level of adhesions instead of distinct two types, and how those varying level of adhesions can create a clusters of cell. For example, we can see here all the red cells are together, all the yellow cells are together, and because they have more cell adhesions among themselves than interniche adhesions. So we can see, dynamically vary these values and see how the clusters formation take place and what are the results we observe. I'm just looking for people who are good in coding because this is mostly a coding related work, and preferably people with same time zone. That's it.
So
These
I
are
had
the
one or two questions here.
yes.
So you want to switch from a situation in which the adhesivity is defined in the XML to something where each cell has its own adhesivity, or you're going to leave the cells
Yes.
with a shared adhesivity?
Different like if I have defined 16 cells of same type, then I want that at 0 MCS all cells will have same
Right.
adhesion strength, but as the MCS proceeds, maybe
Right.
due to the environmental factors or maybe due to another biochemical biophysical cues,
Right.
those adhesion strengths dynamically vary for each cell.
Right. Right. So Gilberto has published a paper a long time ago where he does evolution in a tumor where the adhesivity of each cell changes on cell division so that you can get every cell having a different adhesion constant. So that's something you might want to talk to him about how to do that.
Okay.
Of course, Yi Zhang has published some papers on leader follower. In this case, are you defining leaders and followers to begin with, or is there a dynamic change of leader follower identity?
I want to observe leader follower behavior in a form that the leaders have more cell ECM adhesion and followers have more cell adhesion.
Okay, so you're not going to use something like delta notch to distinguish leader cells from follower cells.
No.
I'm just I'm just trying to understand what defines what defines a leader cell or a follower cell? Just the adhesivity, or is there some other state of the cell that defines that difference?
Basically, what I understand about leader cells that are the one who create a pathway by degrading the ECM
Right,
for the follower cells to go from primary sites to a secondary site, because initially epithelial cells have a square shape. They can't penetrate through ECM unless and until they transition to mesenchymal.
right. So are you planning on having the cells actually secrete a matrix degrading protein?
Yes, that's what's happening here. This is from a published study by my
Okay.
senior. So
That sounds fine. Having somebody else's prior model to work off of is always makes things easier. So I think that's great.